Hi, everyone.
Hi.
Hi, Joe. Hi, Alex. Hi,
Hi,
Tongji.
Tina.
Just bouncing between rooms now, just making sure people don't have any technical issues.
Super.
Okay. Have fun.
Thanks. Good. I'll see, because John should join us too, unless John's bouncing around to the other rooms. So feel free to bounce around too. We're going to probably have a whole bunch of windows open, but I'm happy to moderate. Any questions, I'll take a look through the chat or raised hands. So yes, if any of you want to have more information or want to learn more or want to help participate in any of the focus groups or experiments that we're doing, this would be great. Anyone want to open up and ask any questions? Well, we can also potentially start on what are some of your interests. What has brought you to breakout room number 3? Maybe that'll help. We don't have a list of like set questions. It's just like, let's get to know each other's projects if you're interested. So maybe you could, oh, hi, John. You could tell us what your, are you an original partner on GenFish? Are you just an interested party? And what are some of your interests with the decision-making toolkit? So I was going to start maybe with Emma. Hi, Emma.
Hi. Sure, I'm happy to jump in. So yeah, I'm Emma Sharkey. I'm actually on the Research Oversight Committee for the project. So I just wanted to come to the meeting, see what's the updates are, see what people are saying, and just kind of learn where things are at with the project. So yeah, and my interest in this area, my background is in Indigenous relations, so particularly kind of looking to see what kind of participation there is for Indigenous communities and what their thoughts are on the work so far.
Excellent question. Amy Tongji, do you have anything you can add at this point? Or John?
This is Amy. On the topic of Indigenous communities, my PhD student who's working on the project with me is going to be focusing her dissertation on Indigenous communities and issues of food security and how all of the GenFish project, the eDNA potential, is perceived, and especially in the larger context of environmental challenges in communities. So trying to better understand what potential issues of concern may be identified, and what GenFish can do in response and helping with mitigating those issues.
Mm-hmm.
And so Tina, if I may jump in, actually, my master student Maya is in the meeting room, and she's going to work on Indigenous versus corporate fisheries conflict, and see how the conflict can potentially be mitigated by better technology, better communications, and so forth. And Maya, if you are there, feel free to talk.
Yes, welcome, Maya.
My name's Maya. Yeah, I'm a first-year master's student working with Tongji, so it's very beginner level, but my interest does strongly lie in Indigenous fisheries issues, kind of with the conflicts that came about this summer in Nova Scotia, and I hold strong interest in behavioral economics, so I'm really looking to see the cooperation between the two in that way, in decision making. Yeah.
Great. Thanks for sharing. I also know that when we were working on our paper on actualizing transcriptomics, we had as one of our partners in case studies just a member from Chippewas First Nation, and she spoke on behalf of just Indigenous communities, not particularly her own, but she said in terms of actualizing these for the transcriptomic or the fish health toolkit, it was really important to first build trust. Trust was I think the number one best practice going forward, and then came training. Training issues was another really important outcome in terms of how do we actualize and operationalize these chips. And so it was building trust, it was training, and then it was also data storage and some more of the practicals coming out of that as well. So that was just some of the takeaways that we're getting trickling in on within our gels section here of GenFish. Steve, thanks for joining us. We just had a question from Emma from our ROC. Hi Emma again. And she was just wanting to know what, anyone has been engaging already with Indigenous and the concerns that have been being expressed. So Amy already mentioned that she's got a student who's on those issues. Tongji said with her own student, Maya, who's actually here with us and engaging and looking at the toolkit as a type of use in conflict mitigation. And Steve, and did you have anything to add on that as well?
Yep, working with Andrea Reed, who is now the leader of the Indigenous Fisheries Research Unit at the University of British Columbia, who happened to finish her PhD in our lab just a few months ago. She and I and a few other folks just scored 100K from the Great Lakes Fishery Commission to focus on, and that's just the first step. That's just for the early engagement and relationship building phase that will then lead to some empirical social science research around understanding Indigenous perspectives on sea lamprey control and sea lamprey and fisheries management in the Great Lakes. So the sea lamprey is really the focus of that. However, we're going to be able to piggyback off of those consultation and relationship building phases in year one, and then be able to slip in a few questions. Again, it wasn't a huge focus of our proposal, but this is just a synergistic win that's come along with the right people, Andrea Reed leading it, and we can work with her, given her expertise in that sphere.
That's fantastic. Leanne, your hand is up. Hi, Leanne, how are you doing?
I'm good. How are you? It's great to sort of put a face to so many names. As an end user, technically, I guess, on this project, partners, it's been great hearing the presentations, and my mind is spinning with ideas. So just to introduce myself, I'm Leanne Postma, and I'm a science manager with Fisheries and Oceans, and I'm at the Freshwater Institute in Winnipeg. And we have countless projects and programs, and so that I see these tools, all of these tools, all 3 or 4 of the tools that you've described being useful in a number of the initiatives we have and the work that we do. I myself really, as a manager now, bridge that sort of that transfer of knowledge from the science programs to sort of managers and decision makers within the government. And so there's a lot of questions I have about that, and a lot of information I think is going to be really useful coming out of your project. But on the pure science program side, I'm really intrigued by the work you're doing with Indigenous communities and the transfer of knowledge and information and engagement with Indigenous communities in the area of eDNA and this particular application of it. So, we have a program, for example, that is just starting up related to the expansion of the Trans Mountain Pipeline in Alberta, and working with all the Indigenous communities and organizations along that pipeline route, looking at cumulative effects on fish in freshwater systems that could be affected by the development of this pipeline. It would be really cool to be able to connect with you guys and the information you're gathering sort of at these early stages, because we're in the early stages of our engagement and co-production of monitoring programs for the impacts of this pipeline development. So I could see where we could work together, maybe incorporating some of this information in that program. So just, this is a great networking opportunity for all of us, and certainly I'm looking forward to the conversations we can and will continue to have, hopefully.
That is great, Leanne. John is on, he's chairing, Johnny Villenois, who's here, is chairing the Ethics and Implementation Committee too. Leanne, are you, one way to keep engaged is to potentially be on one of the committees of Fishcast. GenFish, sorry, I got too many. GenFish, sorry, that one. So, and again, you'd be consulted and you'd have your finger on the pulse of what was happening. So potentially we can consider drawing you in a bit more of an engaged role. Not taxing, sure. But it'd be something
and that would be just fine. And I noticed on the participant list you have Eva Enders, who's a research scientist in my group, yes. Also involved in this, and she's actually our lead research scientist on this Trans Mountain Pipeline expansion project. So I think her team would be keenly interested in that type of information too. So for sure, put me on your list, and we'll figure it out.
That's fantastic. Thanks so much, Leanne. John, your hand's raised.
Oh, thanks, Tina. Slight change of topic, so I don't want to stop the flow of this topic.
No, we'll come around. It's a free flow. Yeah.
So it's okay. Yeah, I'll throw this out as a question to the group. So yeah, a great summary you did, Tina. By the way, that was wow. Made it sound much better than what I could do. Anyway. So I'm working on 5.3, which is, about the benefits costs of adopting the toolkits versus using the sort of conventional technology. I have, as you indicated in your summary, that's an alarm because I have to give my dog her medication, but I'll get to that in a moment. Yeah, much of this work can't really happen until the toolkits are close to being developed, but a bit of literature review anyway has turned up. I've found, I think, 3 or 4 papers which attempt to measure what they're calling the cost efficiency of eDNA sampling as opposed to conventional capture sampling. The most recent one I've found is 2018. So my question to the group is anybody aware of any ongoing work, unpublished yet, working papers where people are doing this sort of thing, trying to measure the cost advantages of using eDNA sampling versus the conventional way? Just don't want to miss something that may be more recent.
So I do know that Daniel Heath, he has one student, a master's student working with him already, and who's going to be part of his master's thesis is to contrast. As part of it is looking at, okay, what is the cost efficiency of doing, and I think he's doing 100 fish species with eDNA, and again, contrasting that with conventional methods. So what we try to do as well is within our activities is we have a list of questions, because a lot of us in the social science and in the GEL section, we have a lot of potentially unknowns, kind of the information we need to be able to engage with our end users. And so part of that is, a lot of it was, okay, a primer on a genome, a primer on how much does the chip cost? And it was great getting Daniel to say $500 and potentially even less. And that's the kind of information we need. And so, John, I would recommend popping over into breakout room, I would say number 1, and asking, because Bob Hanner would also be incredible to help you out with that question. But what I'm going to be doing is making a suite of these questions potentially, and so that we can have that free exchange going between. Because I know, John, that's one of I think the main levers of this decision support tool is cost efficiency. And there hasn't been a lot of studies that we've come across. And just to amplify Amy's work that she's doing with Sapna and with Steve and a larger one of us, it's like asking expert in the field what questions are needed to be addressed so that we could adopt these tools. And one of them would be, we need to have more quantitative metrics and measures of cost efficiency. And I could see that as being one of the questions that emerges, John. So I made note of it, and it's something that I would recommend. If you need to pop out first, pill your dog, medicate your dog, and then, come back into the group and let us know what you find.
I'll wait until we break at 3:30. She can last until then, I think. But that's helpful, Tina. From what I've been reading, many of the costs occur in the field, not the cost of the chip or the cost of running assays or whatnot. But it's the time spent out there in the field collecting samples or electrofishing or whatever. So, and I do know Bob quite well.
Oh, good.
And it sounds like Nick Mandrak is somebody that we should be talking to as well, because he'll be doing, I guess, the field validation. And that's, I think, where a lot of the data will have to come from.
Good. Yeah, we'll get on him. And again, we'd have to check the budgets. I don't want to speak for the budgets, but to get students on it as well, so that we can have students giving them the opportunity to potentially take that on.
Oh, I'm not going out in the field.
There you go. We can start advertising for students for you, John.
Too many mosquitoes.
No, I know. Thanks, John. Steve, I want to address that you have in the chat. Yeah, please tell us about your Pan-Canadian Management Survey. Go for it.
Cool, and it kind of fits in between the cracks, yet connects some of the pieces within within activity 5. And there were parts of this that weren't explicitly in the proposal, but at the same time, we kind of needed to ask. So where is fisheries management today? Where have we been? Where are we today? And where are we going? Which will allow us to contextualize these newer tools and technologies. And so Andrew Howarth, who I believe is on here right now, he is a PhD student working on that. And so he has already surveyed, I think, just shy of 40 fisheries managers and practitioners in provincial, territorial governments, and also hitting on some of the DFO folks, not in the science pillar, more frontline staff. So some of the fisheries protection program staff, restoration staff, and so on. So there are two. There's one territory and one province which are being a pain in the butt to find people, but otherwise, Andrew's done a great job. So there are specific questions in there about these technologies, but it also just gives us an overall feel for perceptions about technology and barriers, and so it fits into the knowledge mobilization space. As well as some practical things, I think, that'll guide us with this toolkit. And you'll hear from Andrew for one slide for 1 minute later.
Yeah, thanks for setting it up, though. So it's good. Tongji set up Maya, you're setting up Andrew. Amy, I did see your hand up, but you took it back down. But did you have anything you wanted to add?
Yeah, I was just going to add that there's obviously a lot of overlap with my interest in the ethics side of things and what John's going to be covering. And one of the questions that I've had that I really haven't found a good answer for yet is the number of fish that would be killed in traditional methods versus through these new methods. And, because the cost-benefit analysis isn't just an economic one, but also an ethical one on the number of animals that are, target animals, but also, other animals that are taken as a result. So, John, if you find any answers, let me know, because it's something that I haven't really been able to find a concrete answer for yet. And, perhaps because there just isn't one yet.
You raised a really interesting point because, and Steve mentioned, we do have like an end user committee, and we're going to try to disseminate these types of questions through the end user community, because again, it'll probably be context dependent. You might have some that are definitely, some of the partners that I'm working with, they're doing satellite telemetry. So I know they're already doing operations on the fish, but they're really hesitant to take any lethal samples whatsoever. So for them, it's really important not to kill fish. And that's for the fish health toolkit. And you have others that are doing sampling anyways in terms of surveys. And so for them, it's not as much of an issue. So it lies along a spectrum. And that's why hitting like our different partners on the group would be fantastic. And so being able to reach out and make a call if you want to do focus groups or one-on-one interviews, I think that would be a fantastic thing to do. Yeah, go ahead, Stephen. I know we have some comments also. Oh, you already have a comment in there.
Yeah, there's certainly some trade-offs there too. So for example, we can use gill nets to capture fish for biological sampling, and that's a great way to figure out what's there. But it also gives us other information that eDNA does not. It tells us about the size structure of the population and growth rates and diet. So if the question is simply how many fish are there, and one is normally going to use a lethal technique, then certainly eDNA is going to cut down. But there is also loss of information if one needs that other stuff too. So what else was I going to say? The other thing is, even the more benign things we use, like say a seine net or a backpack electrofisher, which have been refined and are generally regarded as sort of low impact activities, it's not a free lunch. There, some fish will die. There's certainly stress and injury. You can't handle a fish without stressing it, and you can't catch it without injuring it. That's just, so there's also that sublethal welfare component that we could talk about as well.
If I could just jump in.
Go right ahead.
Tina, I would just, well, there it goes again, because I haven't fed her yet. I would just, thanks for that heads up, Amy, and I'll keep my eyes open for that. I would just say that, proper cost-benefit analysis would not exclude ethical costs, as difficult as they may be to measure. We would not distinguish between economics and ethical costs. They should be in there, definitely.
Fantastic. All right. Any other questions for our group?
I better go give the dog her pill. I'll see you back in the main meeting.
Excellent. I just also wanted to make a point that although our the GenFish grant, as Daniel mentioned, was one to enhance, let's say, the economics of Canada through fisheries, we do have a section in there on species at risk, because environmental consultants are really interested in this as well, because as soon as we discover a species at risk, that will impact how much we're allowed to harvest, and it has like knock-on effects in terms of commercial fishing or recreational fishing. And so Amy, when you're talking about the ethics of sampling, especially if they're species at risk, which are part of our grant because of the connection that they actually have to shutting down commercial fisheries and recreational fisheries, which is huge, that definitely would be a non-lethal sampling that would be desired. And so that's why I think one of the great interests, I know eDNA, obviously non-invasive, but even the transcriptomic health toolkit, if we could do a muscle biopsy, or let's say just a bit of a gill arch sample without it being lethal, but can basically tell about the health and the status of the individual, and then make potentially extrapolations to the population, would be really, really powerful and something we would try for. I just have a quick question. So Phil Karpovitz, are you still with us in our breakout room? Because I do have a question for Phil. I saw him before. Okay, well, he's also part of GenFish. I don't see he might pop in, but it's something we might be interested in, Amy, with your media sweep, because you're looking for terms like transcriptomics, gene expression, and Phil works directly and he's familiar with CRISPR technology, so gene editing technology. So he would be a really good person to hit up too if there are any extra terms to do your media sweeps with that I could have not been aware of. We're trying not to do CRISPR gene editing in this, but we're doing things through broodstock selection, and it is, like GMO fish over time, just through our selective practices. But in terms of gene expression, he would be someone who would be really able to provide and shed more light on that situation too. Oh,
thanks. I appreciate that, because when we did our search with the keywords that you had given me, it net about 250 hits over the past, that's 20 years, looking at the New York Times in the US and the Globe and Mail here. Yeah. So, maybe he can give me some suggestions to just cast the net a little wider and see, might end up with things that, aren't necessarily exactly on point, but it would be helpful so that we make sure we're capturing as much as possible.
Excellent. I did ping Phil if he might be with us now.
Yeah, I'm here.
Oh, yay. It worked. I felt like I summoned you. That was amazing. The power. Phil, I was just talking to Amy. Amy's doing a media sweep of some words to Google that might give her some hits. You know what, Amy? I'm going to let you do an even better job explaining it. So, Amy, Phil, Amy, go if you don't mind, Phil. We need your expertise.
Okay.
Hi. So I'm we've done a media analysis on the eDNA portion, and now we're moving on to doing a media analysis of the second objective of GenFish. So looking at the transcriptomic side of things.
Okay.
And so Tina had very generously given me a number of keywords to search for. And so I was just saying that we've gone through, and with those keywords, we've found about 250 media articles over the past 20 years in Canada and the US, and New York Times and Globe and Mail. So I'm wondering if there are other keyword terms that we should maybe be looking for that might be used in the media.
So for example, I gave examples of gene transcription, gene expression, transcriptomics. If Amy had put in CRISPR, it would have lit up, definitely. So there are those contentious ones, or gene editing would have been potentially another one. But that would almost be biasing towards, you know, that, and that's not what GenFish is about. But can you think of any other keywords in the media that would be used to describe what, looking at, let's say, genomics?
Mm-hmm. I mean, there's a lot of ohms, right? Like transcriptomes, but you've already got that word. That's really the closest one I can think of that would be describing what we're doing here.
Okay.
You know what? If you did mRNA, you'd get a whole bunch of vaccine ones right now.
Oh my God.
But mRNA is one. It's legitimate, right? Because that's what's being transcribed.
Yeah.
Okay.
Yeah.
The RNA.
Yeah.
I don't know. The transcription is that's your main term, right? A transcription, transcriptome.
But when we're doing lay summaries, Phil, what do we write about? Yeah. When we give public talks.
Yeah. I don't think you're going to find a better one than transcriptome. I can look, but I'm not really sure that there's much else to really describe it as.
Yeah.
Well, I appreciate that. I just want to make sure that, you know, I'm being as exhaustive as possible.
Yeah. The only other one I could think of is translatome.
Okay.
Because that's transcriptome is bigger than translatome. It means everything that's expressed, means that all the RNA that's made, and the translatome would be all the gene products that are made, so all the proteins that are made from that RNA. So then, you know, the mRNA is really like more representative of the translatome, perhaps.
Thanks, Phil.
I can give you one more.
That's
awesome. I don't have that, so thank you.
Okay. I can now make you, if you need to, disappear. Go ahead. But thanks for coming in when I called you. Tongji wants to just be kept updated on the cost-benefit analysis, John. And so any informations, and that was a great question about complexity and uncertainty being included is really helpful. Those were some of the questions we had. I don't know if John's still on, if he joined us again. When we had the first pass, when we were writing GenFish, we did have some questions even in previous iterations of this grant when we were writing for Discovery Frontiers. It was, okay, what is our validation even for, not just for the eDNA, but it's also for the transcription part. So in terms of how much gene has to be expressed for us to conclusively say this is probably going to have a downstream impact. And so sometimes there are papers on that in terms of the fold change, and it gets a little bit more, it gets into the stats and the analyses of the data. So sometimes we could have some delta fold change of 2, and that could be considered enough to say this probably would have an impact. So if this DNA got transformed into RNA that then became proteins, and the proteins affected the health, right? Because that's how it goes down the line. Yeah, it would probably end up in a stressed animal. And so that was one of the questions that a lot of our end users are curious about as well. Is validation not just for eDNA? Yes, are there fish here? What guarantee can you give me? But it's also, what guarantee can you give me that this fish would be sick? This isn't indicative, because again, the whole fish might appear healthy, but if it's starting to give signals, this health toolkit is not just diagnostic. It could diagnose what's happening, but it can also act as being prognostic. That if we do have some genes that are worrisome, it's something to be aware of. because this normally wouldn't happen, and there might be some long-term consequences. We might not see it immediately, but it might hit it down the line, either reproductively or after some subsequent generations. So I just wanted to put that out there as well, that there's also that kind of validation and uncertainty that's involved in the health toolkit too.
Thanks, Tina. That helps. And if I am back, if I could just jump in, Steve. Yeah. Steve, I think you sent me a message in the chat. Thanks for that. I was trying to upload a file to you. I don't know how to do it, so I'll email it to you later.
Okay.
So what I was trying to get at when I was asking the question about type 1, type 2 errors is, again, this is coming from an economist, so it may make zero sense to you, but it seems to me if you're using conventional capture techniques, there's still a risk of type 1, type 2 errors. I go out, I electrofish, and maybe I didn't find anything. That's not because there's nothing there. I may just not have found something. So there is an error factor. When we do eDNA sampling, same thing might happen. You scoop a bucket of water out of the lake, and you may not, catch some of the DNA that was left by a sturgeon that was at the other end of the lake, whatever. So both methods are going to have both false positives and false negatives, I would think. What I was trying to get at is whether in this research, in this big project, there are going to be some, quantitative measures so we can, that's going to be part of the cost benefit, right? Is are you treating the uncertainty of the sampling technology? So that's what I was trying. I probably didn't express it very well, but that's what I was trying to get at.
No, and Steve, if you want to talk about the eDNA, but I feel more comfortable talking about the transcriptomics. We will have that kind of error bar, if you will, associated with that. And then the job is to then translate that into type 1, type 2 kind of errors, because we've got the statistical error, but then we want to know, okay, does this mean the fish is going to be sick, yes or no? And that's something that's going to be really interesting to hear from our end users. How much do they want us to interpret their results? How much do we want, should they be contrasted? Because I don't know if we'll ever say you're going to get a 60% chance of misidentification with this technique versus a 40% in that technique. Because it might end, I don't know if they'll be using the same currency at the end of the day, especially with the health as well. Because when we're doing fish health, we will have to, conventionals, you sacrifice it, you do necropsies on them. Versus, okay, well, that could be definitive, but it's usually at the end state. The animal has to appear so poorly that's why you've got a necropsy because it's already dead, or you're sampling it because there's a signal there. Versus the health toolkit, which could act prognostically. And so again, it'll be really interesting to find out what the level of acceptance would be, or what is the level of uncertainty our end users are willing to accept with these tools. And again, it might have to be the way we convey it and relate it to them might not be on the same currency. It's really interesting. I don't know the answer to that, but it's a really, really good point to raise. I don't know if I've actually addressed that comment that you've made, and Tongji is going to come in and rescue me. So go for it, Tongji.
No, Tina, I was just going to say it is actually part of my research interest to look at people's trade-offs on uncertainty and risks and ambiguity, right? And I don't have an answer yet, but so that's exactly why I wanted to know how to parameterize my experiments, because I need to know a rough range of the uncertainty and the perceived risks and the ambiguity in order to put everything in an experiment, and then using that experiment, I can see how people make trade-offs and what is the most likely acceptable levels. So yeah, that's precisely why I'm asking that question. Thank you.
Well, that's great. So Tongji, let us know when you're ready to start parameterizing your models for that, or your experiments, so that they're within likely ranges, right? Yeah, amazing. Okay, well, there's a break until 3:50. You can take a break right now, do some stretches, take a health break. You can stay on and we can continue discussing. I'll be here till 3:50 anyways to field any of your questions you might have. So, let us know. But for those of you that are staying on, I can give you just a brief before this paper is almost ready to be out and submitted. The semester's just been pretty hectic, but I could summarize for you out of our case studies what were some of the barriers that needed to be overcome. And if anybody wants a copy of it, please let me know. And Leanne Posma, who she might still be with us, might be wondering why isn't it out yet, but it is on my list of things to do. She was one of our end users and contributors and co-authors on this paper. But there were risks that people had expressed amongst our partners. And the first was the reliability of having a universal transcriptional profiling. And is it universal across space, across different geographic areas? Is it universal across time? So for example, if you're looking for temperature stress in the spring, is it still going to be the same kind of stressors you're going to recognize in the fall? So there are those kind of questions. Another concern was the need for quantifiable evidence for return on investments of the time and money with measurable improvements. So there's that ROI that's really needed in terms of fisheries and fish culture yields. So again, all about the commercial aspect of it. A third one was a general acceptance across multiple jurisdictions and knowledge holders. Will the results be accepted? And if, let's say, an environmental consultant is using our chip, what happens if the government in its stead says, we haven't validated it ourselves, we're not going to actually rely on your results telling us, well, we can either condone this development or condemn this development. So Leanne, that might be really an interesting point for you as well, what's coming out of this from what you've said. Another one was big data management and accessibility requirements. Who gets to own the data? That's the sovereignty over the data. Again, which was a very big concern for Indigenous communities. And the knowledge and the knowledge holding and the data holding would be with our managers, would be with our communities, not with the scientists themselves. And again, there was an integration and incorporation of knowledge in terms of train us how to sample that you need, train us how to do eDNA sampling, but this paper is mostly for the health toolkit. How do you do then sampling fish without killing them potentially? And that was really important, as well as infrastructure. I need freezers all of a sudden. Are you going to supply me freezers? This is extra. So there are all these hidden costs that need to be aware of. Yeah, and then the last one was incorporation into training, like I said, and best practices. Go ahead. Yes, I think I'm done. I just want to make sure I get all the chats. And that's it. Oh yeah, Joe, go ahead, break in.
Thank you very much. What did Steve just post? I saw he was typing.
Steve's taking a health break.
Yeah. See you at 3:50. Perfect. Okay, so we're at the point now with our 20-minute break. Everybody can just kind of relax, and at 3:50 we will be back. For right now, if you will click leave and rejoin the main room, I'm just going to do a little wrap up, and I will post the link to the informal session.
Okay. Bye everyone. Thank you so much for showing up, and don't forget the informal session afterwards where we can listen to all what our students are doing.